Thank you very much.
Thank you, Claire, and thank you everybody for staying. I had exactly the same slot at crop protection in southern Britain here a fortnight ago, and half the delegates had gone, and I was getting a bit of a complex. So this is good. Yes, 50 years, in fact, hence golden years of monitoring. The Rothamsted Insect Survey celebrated its 50th anniversary 29th of April this year. I'll say a little bit about that at the end. But what I'm going to do is, first of all, say something general about aphids and why it is that they are such important pests, including their role as virus vectors. Then say something about the means that we use to monitor and forecast them and the work that's being done at Rothamsted on insecticide resistance, which is the background to how that all feeds through to the Aphid Alerts, which hopefully a lot of you are aware of and use. And then at the end, I'll just give a few thoughts on what may be the situation 50 years hence when the insect survey is 100 years old. So, aphids, why are they such important pests? Well, first of all, what are they? They're not just green fly. They literally come in all colours of the rainbow. There's no cheating here, no colour enhancement. There are something like 600 species of aphid in the UK, most of which are not pests, most of which are confined to a very narrow host range. But, of course, the group as a whole has got a bad name because of those that are such very important pests. And one of the reasons they're so important is that they're very highly adaptable. They respond to environmental cues extremely quickly. So what we have in this picture here is an adult winged female and an adult wingless female. These aphids are all parthenogenetic, i.e. they reproduce asexually without the need to mate for at least part of the year. And the nymphs, which you can see underneath, may have been produced either by the winged one or the wingless one. You can't tell by looking at them. But the key thing is that those nymphs aren't determined as winged or wingless until about the second of 4 nymphal generations. And whether they become winged or wingless depends partly on the nutritional quality of the host plant, and if that is poor, then a greater proportion will become winged because that allows them to fly off and seek alternative hosts. And also, if the parents or the nymphs themselves are crowded, that will induce a larger proportion of winged forms to be produced because, again, if they're crowded, chances are that it's not going to be long before the quality of the host plant drops off very considerably. And it may only be, a matter of 3 or 4 days between those cues being sensed and the adults sprouting wings or not. So they're very, very responsive to the environment. But perhaps the biggest reason that they are such important pests relates to a phenomenon illustrated here known as telescoping of generations. And what you've got is an adult female aphid. Inside that, you've got a nymph, an embryo that is about to be born. And inside that embryo, the next generation has already been ovulated. So you've got the mother, the daughter, and the granddaughter all there present in a single individual. And that precocious embryonic development means that almost as soon as they become adult, they're ready to give birth. It may only be a week or so between being born and being adult themselves, and in the following week they could easily produce 50 or so offspring. And you don't have to multiply 50 by 50 many times before you've got sort of really large numbers of aphids. So, it may just be a matter of a week or two before they're between them being almost impossible to detect and there being a real serious infestation. And I've calculated that in a single year, aphids that are continuously parthenogenetic, and some are, can get through about 18 generations under typical conditions. And if all of the offspring of every generation survived long enough to produce all of their offspring, after that 18 generations, i.e. 1 year, you'd end up with a layer 149 kilometres deep covering the whole surface of the Earth. Now, Clearly there are many reasons that we're not wading knee deep and worse. I mean, aphids work by producing huge numbers when times are good and then suffering huge losses when they're seeking hosts, etc., etc. But what this does show is how they have such strong potential to be pests. And it also explains why they're very, very sensitive to changes in climate. Warmer conditions, more generations, and that layer would be even deeper. So there we are, there's parthenogenesis in action. And many aphids, indeed most aphids, interrupt that parthenogenesis once a year with production of a sexual generation, and they do that. They respond to the length of the night. The increasing length of the night is what they measure, and then parthenogenesis gets switched off and they produce sexual morphs, and there you go, tonight's the night, and that results in the production of winter eggs. And these eggs, the advantage to them of producing winter eggs is that they are very, very cold hardy, whereas the active individuals are not. And I'll come back to that just a little bit later when talking about the monitoring and the forecasting. So very often the eggs are produced on a completely different set of host plants to those on which the asexual generations are produced, and some of them are well known, such as the black bean aphid, for example, can only produce eggs on Euonymus europaeus spindle. One of the most important aphid pests are carrots, can only produce eggs on willow. Then there's the famous bird cherry-oat aphid and the peach potato aphid, which describes their winter and summer host plants respectively. And then there's a whole load of other rather bizarre combinations of winter and summer host plants listed there. And there's no obvious rhyme or reason as to how those combinations have developed. It's a sort of accident of evolution. There's no obvious chemical similarities between winter and summer host plants, but that is the situation. I do sometimes get asked, if, for example, I'm growing beans and I've got a load of spindles around the place, should I get rid of them? The answer, to be honest, is no, because aphids do fly very long distances, and getting rid of local winter hosts probably wouldn't make very much difference to your own local crops at all. So virus transmission, that is why, probably the biggest reason that aphids are important in arable crops. Just a picture there of aphid stylets penetrating a plant and reaching the phloem. So the viruses that we're mainly interested in, HGCA, barley yellow dwarf and turnip yellows, both are persistent or circulative viruses as they're known, which in some ways is a good thing, because persistent viruses can only be picked up from the phloem, which means that the aphids have got to commit to a phloem feed, which means they've actually got to be aphids that feed on the plant, whereas non-persistent viruses, I'm sure most of you know this, reside in the subepidermal cells and can be picked up and passed on by aphids that are simply probing to find out whether they're on the right host plant, and will disappear if they're not. So non-persistent viruses are very, very difficult to get at with insecticides. Got to be really, really quick. But persistent viruses, at least provided there's no insecticide resistance, are possible to deal with. So what happens is that the aphid puts its stylets in, like we saw in the previous photo. The virus is drawn up, it goes through the gut, it reaches the hind gut, it leaves the hind gut and goes into the main body of the aphid, the hemocoel, from where it works its way to what are known as the accessory salivary glands, and it's from those that it can get pumped back out into the plant, into the phloem. So that whole cycle takes a matter of 2 or 3 days maybe, so there's a sort of a latent period between an aphid acquiring the virus, during which it is unable to put it back into the plant, because the virus has to go through that cycle. So that's the sort of mechanism for virus transmission. And the ones that we're really interested here are turnip yellows, transmitted by peach potato aphid, Myzus persicae, and barley yellow dwarf, transmitted by, on the left there, bird cherry-oat aphid, and the wingless one in the middle with the black cornicles is the grain aphid, Cytobia navini. So what I'm going to do now is say how we set about monitoring these aphids and monitoring and assessing the virus status and whether or not they're resistant to insecticides. And there are two sort of main methods used. One is a network of yellow trays, and under HGCA funding, those are, AHDB funding, I should say, those are operated by FERA. BBRO have a similar set that they use in sugar beet. And we in the Rothamsted Insect Survey use these giant upside-down vacuum cleaners called suction traps. So there's never one perfect way of monitoring aphids or indeed any other insect. So every system has its pros and cons. With the yellow water pans, they're cheap, they're versatile, they're easy to use, they don't need electricity. They only monitor what's going on at a very, very local scale. The field next door may give you a completely different result. So that, for some purposes, is good. For some purposes, it's less good because it means you can't possibly cover the whole country. They are selective insofar as different aphid species are attracted differentially to yellow. So whilst you can compare what's going on with a particular species in space and in time, you can't compare numbers of different species just because you get 10 times as many potato aphids as peach potato aphids, say, in the traps, doesn't mean to say there's 10 times as many going into the crop because they're differentially attracted to yellow. The location of the yellow traps varies from year to year. The time series is relatively short, certainly compared to the suction traps. So the suction traps stay in one place. They're big things. They're not going to be shifted around. The time series is long, as I say, 50 years for some of the traps. Anything that's unfortunate enough to fly over the top goes down the tube, so they're relatively non-selective. They are regional in their nature. Which in some ways is good, in some ways is bad. It means that with a fairly small network of traps, you can get a pretty good idea of what's flying over the whole country, but there will be a lot of field-to-field variability according to specific characteristics of those fields. They need electricity, of course. The infrastructure's expensive, but, smiley face, the infrastructure is paid for by the BBSRC, and the traps are used for an incredibly wide range of purposes, from the sort of very applied advice giving right the way through to sort of fundamental studies on insect population dynamics and other things. So there are currently 15 suction traps around the UK. As from this year, there will be a 16th at FERA. There used to be one at Askham Bryan. For various reasons, that got closed 3 or 4 years ago, and this one kind of fills that gap. So that's the last thing an aphid sees before it disappears down the tube into a pot of alcohol, and that's the first thing that the aphid identifiers see when the samples are emptied out onto the bench. There they are, wonderful team of people, and their job is to separate out the aphids from the other insects, identify the aphids, get the, which is done with the help of an in-house key, get the data onto the computer for purposes of fundamental and applied science. Now, as well as just counting individual aphid species, it's sometimes necessary to know what's going on at a level below the species, and that is particularly the case with bird cherry-oat aphid, because that's an aphid that has got both life cycle types open to it. Some clones respond to the increasing length of night by producing sexual forms and flying to bird cherry where they lay eggs, and those will play no part in spread of BYDV during the autumn and winter, whereas others do not respond to increased length of night. They carry on reproducing asexually, and it's these that potentially can go to cereals and play a role in BYDV spread. So when you look at our newsletters, you'll see that we report on the proportion of those at Rothamsted, at least, which are of each life cycle type. Unfortunately, the winged forms from the two life cycle types are morphologically absolutely identical. You can't tell by looking at them. But we've got a second trap at Rothamsted in which we collect the aphids fresh and alive. And if you tease the embryos out of the forms that are potentially going to cereals, which is the ones on the left, they are a kind of pinkish colour, whereas the ones that are going to go to bird cherry on the right there are a kind of greeny yellow colour, and that's how we know what proportion of what we're getting in the suction traps is of each life cycle type, and the proportion of asexual forms tends to be higher towards the south and the west and lower towards the north and the east. And we're also able to give a bit of an indication the previous year as to what those proportions are likely to be, because there's really quite a nice relationship shown here between the temperature in winter and the proportion the following autumn that are going to cereals as opposed to bird cherry. So if you look at the red 2014 point, if I can get the pointer just there, look at the red 2014 point, the winter of 2013-14 was very mild, and the proportion of the cereal colonisers this year going in this autumn was higher than usual. And the reason for that is that low temperature will select against those active overwinterers because they're not cold hardy, whereas the eggs are. So mild winter first one year is a potential first sign of a bit of an issue the following autumn. So, insecticide resistance, huge issue now, especially in peach potato aphid. For whatever reason, peach potato aphid seems to be incredibly plastic and able to become resistant to almost everything that we can throw at it. So there are 4 major resistance mechanisms now. First of all, the first one to be discovered was elevated carboxylesterase that confers resistance to organophosphates. And what happens there is that gene duplication leads to overproduction of this esterase that sequesters the insecticide before it can reach its target site. Then there's MACE resistance, modified acetylcholinesterase, which is a target site mechanism conferring resistance to the dimethyl carbamates, i.e. pyrimidine. Then a series of mutations in what's known as the voltage-gated sodium channel, and these are known as KDR or knockdown resistance mutations that confer resistance to pyrethroids. And then finally, sadly now, there is beginning to appear resistance to the neonics as a result of a mutation in the nicotinyl acetylcholine receptors. So we're able, using aphids taken direct from the suction traps, to look, we've got diagnostics courtesy of our colleagues in our biological chemistry and crop protection department that will tell us whether the aphids that we're catching have these various mechanisms. Now, this is up and down a lot, but this is the resistance mechanism to OPs, and all you really need to look at here is that in more recent years, the resistance mechanism has practically disappeared from the population. That's because, of course, OPs are no longer used to control aphids, they can't be, and there's a known fitness cost to harbouring that resistance mechanism in the absence of insecticide pressure. So those that have got that resistance mechanism are at a disadvantage when there's no insecticide being used, and so it's disappeared from the population. So OPs would actually work very well now to control Persicae, but I'm sure that if they were used, which they won't be, that resistance mechanism would be back in no time at all. MACE is a very different picture. That's gone up and up in the past 10 years, and now pretty much 95, 96, 97% of Mysis persicae are resistant to pyrimidine. Unfortunately, this resistance mechanism is in the two commonest clones known as O and P, which form the greater part of persicae populations in this country, and so pyrimicarb is useless now against peach potato aphid. Pyrethroid resistance is quite interesting because the diagnostic that we had suggested that that resistance mechanism was disappearing from the population over recent years, but it was very clear that there was still a big problem in the field from resistance to pyrethroids. So again, my colleagues, Martin Williamson in particular, went looking for to see whether there were any other resistant mutations in persicae, and they found what's known as new super KDR, which unfortunately also exists in clones O and P, and again is present in about 95% or more of the persicae population. So not only is pyrimicarb now useless, pyrethroids are now useless against persicae as well. So neonic resistance at the moment does not exist in the UK, but it is there, especially in the peach growing regions of France and other parts of southern Europe. It's only a matter of time, I suspect, before we get immigration from France to the UK. And you can imagine the tabloid headlines when that kicks in. But I think it's only a matter of time before, sadly, neonic resistance is likely to hit the persicae population in the UK. English grain aphid resistance first noticed, at least control failure first noticed in 2011, and a diagnostic was developed for, it's a KDR type resistance mechanism again, and because we keep all of our samples, we were able to look back through past years, and we found that the resistance mechanism was present in Curtin at least as far back as 2007, and the chances are, I should think, that it first appeared maybe 2 or 3 years before that. Then it sort of shot up dramatically. It looked as though it might be heading in the same direction as persicae with 95+% resistance, but it stabilised a bit in 2013. Hot off the press for 2014, those are the resistance levels, so it's remained sort of reasonably stable. So there may be some grounds for optimism that it isn't going to get worse. Trouble is that it may be that there are certain clones of grain aphid that tend to feed on cereals, and it may be in all of those. And certain clones that tend to feed on wild grasses, and it may not be in those. So it could be in 100% of grain aphids feeding on cereals. Quite interestingly, actually, it appears to be in just one single genetic background. It looks as though one aphid developed this mutation, and then in a matter of 5, 6, 7 years, that the progeny of that aphid have come to dominate the whole of the UK. And such is the advantage of having that resistance mechanism. So towards the end, I just want to show how those long-term data can be used. At the beginning of the year, we give forecasts for spring-sown crops as to what the aphid situation is likely to be, and that's based on, which is really, I still think, quite a remarkably strong relationship between winter temperature and the time of the first appearance of, in this case, peach potato aphid. So 2014 was the winter that we've just had, 2013-14, was the 4th mildest in the 50 years that we've been collecting data. We were forecasting early aphids, and we certainly got early aphids. And as a result of similar relationships, we were also forecasting large numbers, and we got large numbers. So winter temperature is a pretty good guide to the timing and size of spring migrations in those aphids that overwinter in the active stages as opposed to an egg, which includes peach potato aphid. So all of this feeds through to the Aphid Alerts that we produce for AHDB. And outputs include things like this. You can virtually, don't ignore the raw data table unless you can sort of really are used to looking at that. It doesn't tell you a huge amount. But what we do is we put out graphical outputs like this that show with the red line for each of the main pest species and for each of the trap sites how this year's population is building up, and you can see that it was very early and large this year. The blue line is the previous year, and if you can think back far enough, 2012-13 was actually a much colder winter than there's been for a while, and that led to very late first flights. And then the green bars are the 10-year means. So you can see at a glance as the population builds up, whether it's dramatically earlier than usual, whether it's dramatically later and you probably need to do nothing at all, or whether it's sort of roughly average. We also produce Aphid Alerts, which kind of just give the main sort of aphid news of the week, as it were, which are just coming to an end now. It's all over bar the shouting unless we get some dramatically warmer periods. The aphid flights have just about finished. We haven't had enough hard frosts yet to kill them all off, but they won't be walking around the crop unless the temperatures get up, 4, 5, 6 degrees C, and then they'll only go slow. So unless the winter is incredibly dramatically mild from here on in, it's probably pretty much over now, and we'll, if anything dramatic happens, we'll put out further alerts to say so. So we really do welcome feedback on these alerts, and we do get feedback from a fair few people, but we could do with a lot more. We're also always very interested in field reports, because unlike Jensen, we haven't got a gang of people able to go out scouting the fields. And it would be absolutely great to get more feedback on what's going on out there. So currently then we produce the forecasts. We tell you about forecasts of timing and abundance in spring. Autumn is a lot more difficult. We're working on that, but it's a lot more difficult. We provide updates as the year goes on abundance, on distribution, on the morphs of bird cherry-oat aphid, like I explained, and on the insecticide resistance status. But what about the future? What do you want? We did actually used to measure the proportion of aphids that were carrying BYDV, but that sort of bubbled along at 1 to 2% year after year after year after year, and we decided that it wasn't worth carrying on with that. Okay, we know, but what do you do with that information? Of course, if there's a sudden change, we won't know about that, but potentially we could go back to looking at virus. As I say, we'd like more feedback from the field. We'd like to know what more information you'd like, and also how it's presented. Should we be moving more to an app type information system? So top left there, I'm just putting in an aphid app for you, but we haven't developed anything along those lines yet, but we potentially could. As to the future, well, I've said that the Insect Survey is 50 years old. We had a fantastic party on 29th of April, and in fact the picture of this cake was tweeted far more than the science around the meeting that was describing the 50th anniversary. But we also had, this is Robin Taylor, who is son of the founder of the Insect Survey, Roy Taylor. He's an entomologist in Texas, came over especially for the celebrations. There he is with a very large butterfly net unveiling a commemorative plaque to the Insect Survey. But 50 years' time, when I will certainly be past caring, but I do care about children and grandchildren, will there be chemicals available to control Mysis persicae and indeed other aphids? There's no a priori reason why aphids that are not currently resistant won't become so. And these things are fairly obvious, but still need saying. We do need to avoid prophylaxis as far as possible to prevent resistance build-up and to deploy chemicals according to risk, hence the sorts of monitoring and forecasting that we do. Risk models and thresholds. Thresholds are very difficult. They always need, they change, they need updating, and it's difficult to fund that sort of research these days. But alternative agronomic methods of control and IPM, as Jens has been talking about, certainly should be strongly supported and encouraged in order to stave off major resistance problems. So that's it, and I just want to thank my Insect Survey colleagues past and present, and SASA as well. SASA in Edinburgh, they run the 4 traps in Scotland. They identify the aphids and send the results to us for inclusion in our bulletins. BBRO, AHDB, very important funders. There are two of our major funders, but I do also want to acknowledge Frontier, Hutchinson's, Bayer and Agri, who also put a bit of funding into the insect survey. But about 2/3 of our funding comes from the BBSRC as a national capability grant. So special thanks to them and congratulations on their 20th year. Thank you.
Thank you.